For hundreds of millions of years, the rule seemed simple. Animals ate plants. Plants captured sunlight. That exchange built forests, grasslands, and nearly every food web on Earth. Plants turned light into sugar. Animals turned plants into motion. The pattern felt permanent. Then, in certain swamps and acidic bogs, something broke that rule. There are plants alive today that trap animals. They close in fractions of a second. They coat insects in sticky glue. They drown prey in liquid chambers and slowly dissolve them. They do not have muscles. They do not have teeth, and yet they digest flesh. This was not a single strange experiment in evolution. Genetic evidence shows carnivory evolved multiple times in separate plant lineages, different families, different continents, same outcome. Something in the environment kept pushing plants toward the same solution. But what kind of environment pushes a plant to turn toward flesh? How does a rooted organism evolve something that functions like a stomach? And if this strategy evolved more than once, can it evolve again? Tonight, we look at the moment the direction of consumption reversed. But before we begin, I'd love to know where in the world you're listening from tonight and what time it is there. If these calm science journeys help you unwind, a like and subscribe truly helps this channel grow. Now take a slow breath and let's examine how plants crossed a line evolution had held for hundreds of millions of years. Long before leaves ever folded inward or secreted enzymes into waiting chambers, the land itself was still learning how to host life. The first plants that crept from shallow water onto bare rock entered a world without roots to anchor them or soils to cradle them. More than 470 million years ago, thin green films clung to damp surfaces, drawing carbon from the air and light from an unfiltered sky. Their bodies were simple, pressed close to the ground, absorbing what little moisture and dissolved minerals they could find. Over vast spans of time, structure deepened, tissues specialized, vascular strands formed within stems, slender conduits capable of lifting water upward and guiding dissolved nutrients through growing cells. Xylem and phloem became internal rivers, allowing height, allowing reach. Plants no longer remained pressed to the earth. They rose above it. Forests eventually followed, weaving shade and humidity into the air. Roots pushed downward, breaking stone into fragments, inviting fungi and bacteria into intimate exchange. Soil, as a living system, emerged slowly from this partnership between mineral and organism. Within that darkening ground, nutrients cycled with patience. Nitrogen and phosphorus seeped from decaying leaves and fallen trunks. Microbes transformed invisible atmospheric gases into compounds plants could absorb. Fungi threaded through root systems, extending their reach in exchange for sugars formed in sunlit leaves. Death fed growth, and growth prepared the next layer of decay. It was an economy built on light and time. For hundreds of millions of years, this arrangement endured. Plants competed by stretching taller, by widening canopies, by scattering seeds across wind and water. Success depended on efficient photosynthesis, on the ability to capture sunlight and convert it into living tissue. When climates warmed, forests expanded toward the poles. When ice advanced, vegetation retreated and returned again. Continents drifted, mountains lifted, seas opened and closed, yet the fundamental strategy held steady. Roots mined the soil, leaves harvested light. The food web rose from that exchange without interruption. Even in landscapes where nutrients were thin, the solution remained incremental: grow more slowly, form alliances with microbes, shed leaves to reclaim internal stores. Adaptations accumulated within the boundaries of that original pact between plant and earth. No traps were required, no rapid movements disturbed the stillness of leaves and air. The world before carnivory was not lacking in complexity. It was structured, layered, and remarkably resilient, a system refined across deep time through cooperation between sunlight, stone, and the quiet chemistry of decay. Above the quiet architecture of roots and leaves, the air has always been thick with nitrogen, an invisible ocean surrounding every forest and marsh. It drifts freely, untouched by most living cells, bound in a form too stable to enter directly into the chemistry of growth. Though it composes the majority of the atmosphere, it remains distant, locked behind strong molecular bonds that plants alone cannot break. Abundance in the sky does not mean availability in the soil. Inside every green cell, nitrogen is threaded through the most intimate structures of life. It forms the backbone of amino acids, which fold into proteins that guide metabolism. It anchors the bases of DNA, preserving instructions across generations. It sits at the heart of chlorophyll, the pigment that captures light and converts it into chemical energy. Without nitrogen, leaves pale, growth slows, seeds form reluctantly or not at all. The entire machinery of photosynthesis depends on this single element moving steadily through living tissue. Plants rely instead on a quieter partnership beneath the surface. Specialized bacteria dwelling in soil and root nodules perform the rare transformation known as nitrogen fixation, converting atmospheric nitrogen into ammonia and related compounds. Other microbes carry out nitrification, shaping these molecules into forms that roots can absorb. This underground exchange is subtle yet essential, a choreography of enzymes and membranes unfolding in darkness. Where microbial communities flourish, plants prosper. Yet not all landscapes provide such generosity. In waterlogged wetlands, oxygen thins within the soil, slowing microbial metabolism. In acidic bogs, decomposition falters, and nutrients become trapped within partially decayed matter. Heavy rainfall can leach soluble nitrates downward, carrying them beyond the reach of shallow roots. Sandy substrates allow water to pass too quickly for minerals to linger. In these places, nitrogen drifts away as quietly as mist. The consequence is not immediate collapse but chronic restraint. Leaves remain smaller, stems elongate with hesitation, reproductive cycles stretch thin, energy harvested from sunlight accumulates without the raw materials required to build new proteins. The imbalance is subtle yet persistent, a tension between captured light and missing elements. Natural selection does not ignore such tension. Over countless generations, even slight advantages in nutrient acquisition can tilt survival. Within nutrient poor wetlands, this scarcity shaped every aspect of plant life. Individuals that found marginally more nitrogen produced more chlorophyll, captured more light, and set more seeds. The pressure was not dramatic, it was constant. Low nitrogen limited expansion, constrained reproduction, and narrowed resilience during climatic shifts. In such environments, the invisible chemistry of the soil exerted a steady influence, guiding evolution through absence rather than abundance, through what was missing rather than what was present. In certain low-lying corners of the continents, water lingers long after rain has fallen, settling into shallow basins where drainage is slow and the ground remains saturated through most of the year. Here the surface trembles underfoot, layered with sphagnum moss and partially decomposed plant matter that accumulates faster than it can fully break down. The air above may be open and bright, sunlight pouring down without obstruction, yet beneath that light, the soil tells a different story. Acidic bogs form where rainfall exceeds evaporation, and minerals are steadily washed away. Hydrogen ions accumulate, lowering the pH and altering the chemistry of the ground. Nutrients that would normally circulate through decay become locked within slowly rotting peat. Microbial communities, sensitive to both acidity and low oxygen, operate at reduced pace. The familiar underground exchange that supplies roots with usable nitrogen and phosphorus weakens. Decomposition slows to a near suspension, and organic matter builds into thick, spongy layers that hold water like a reservoir. Swamps and peatlands share this saturation. Oxygen struggles to diffuse through waterlogged soil, and without oxygen, many bacteria cannot efficiently convert nitrogen into forms plants can absorb. Nitrification falters. Mineralization slows. The result is not absolute absence but persistent scarcity, a thin trickle of nutrients within a landscape otherwise drenched in light. Heavy rainfall compounds this restraint. In sandy or porous substrates, soluble minerals dissolve and move downward beyond the reach of shallow root systems. Each storm quietly resets the nutrient balance, carrying essential elements away molecule by molecule. These habitats are paradoxical. Sunlight remains abundant, striking open wetlands without the dense canopy of taller forests. Photosynthesis can proceed with vigor when leaves are exposed, yet the raw materials required to translate captured energy into growth remain limited. Chloroplasts harvest photons. Cells wait for nitrogen that arrives slowly. The imbalance creates a subtle tension within plant tissues. Energy gathered in excess of the building blocks required to construct proteins and nucleic acids. Traditional strategies, so effective in richer soils, offer diminishing returns. Extending roots deeper yields little when the deeper layers are equally depleted. Forming symbioses with fungi provides some advantage, yet even those networks draw from the same impoverished substrate. Growing taller demands nitrogen that is not present. Producing seeds in abundance requires phosphorus that leaches away with each season of rain. In these bright, yet nutrient-starved environments, survival depends on innovation measured in small increments. Over long stretches of evolutionary time, such conditions act as steady filters. Individuals capable of extracting even marginally more nutrients gain a slight edge in growth and reproduction. The wetlands do not shout, they apply pressure through consistency. Waterlogged, acidic, low in decomposition and oxygen, they shape life through constraint. Within this quiet insistence of scarcity, the stage is set for adaptations that depart from the long-held reliance on soil alone. Along the surface of certain leaves, small, translucent beads began to appear, each one perched at the tip of a slender hair. They glistened in open light, catching the brightness of day without intention or design. At first, these glandular hairs were not instruments of capture. They secreted viscous droplets that discouraged herbivores, trapped windblown particles, or reduced the growth of fungi along delicate tissue. The secretion was protective, a modest biochemical shield spread thinly across the leaf. The droplets contained polysaccharides and enzymes common to many plants, compounds capable of interacting with the outside world in subtle ways. When dust settled, it adhered. When spores drifted down, some were immobilized. The leaf remained otherwise unchanged, still performing photosynthesis, still anchored in soil that offered little in return. Yet within this quiet, surface chemistry, an unplanned interaction occasionally occurred. Small arthropods, drawn by moisture or reflective sheen, would alight and struggle against the adhesive film. The more they resisted, the more firmly they became ensnared. Their movements bent the delicate hairs inward, pressing bodies closer to the leaf surface. Death followed not through intention but through exhaustion. Over time, microbial communities began to act upon the trapped remains, breaking down proteins and chitin into simpler compounds. Nitrogenous molecules seeped outward, diffusing across the cuticle and into underlying cells. The gain was slight, almost imperceptible within a single generation. A few additional amino acids entered circulation. A fraction more chlorophyll could be produced. A developing bud received marginally greater support. In landscapes defined by scarcity, such increments mattered. Plants bearing slightly stickier secretions retained more incidental prey. Those with epidermal cells capable of absorbing dissolved nutrients from the leaf surface made more efficient use of what adhered to them. Natural selection does not require dramatic leaps. It favors cumulative advantage. Variations that enhanced secretion density, enzyme activity, or absorption capacity were preserved through reproduction. Over many cycles of growth and decay, glandular hairs became more numerous. Secretions grew more viscous. Enzymatic profiles shifted subtly toward proteases and phosphatases that accelerated breakdown. What began as surface defense gradually acquired nutritional consequence. The transformation did not happen everywhere, only where soil offered little and light offered plenty. In those constrained habitats, leaves that passively captured organic matter possessed a quiet edge. A plant that absorbed even a trace of nitrogen from an immobilized insect could invest that surplus into root extension or seed production. Increment layered upon increment until the incidental became adaptive. Through this slow refinement, a small advantage turned to evolutionary currency. Adhesive droplets that once served only as protection began to participate in nutrient acquisition. The leaf surface, formerly a boundary between plant and world, started to function as an interface for absorption. Within the steady arithmetic of selection, what was once accidental settled into structure, and structure into inheritance, carried forward as a subtle but enduring shift in how certain plants interacted with the life that touched them. Across many generations, the surface of certain leaves no longer remained flat and passive. Subtle curvatures began to appear along their margins, as if the tissue itself were responding to repeated contact with the outside world. Cells along one edge divided at slightly different rates than those on the other, creating a gentle inward bend. What had once been an open plain gradually formed a shallow cradle, a shape capable of holding more than light and rain. This curvature was not sudden. It emerged through small asymmetries in growth patterns guided by hormones such as auxin that regulate elongation within plant tissue. When stimulation occurred along the leaf surface, whether through mechanical contact or chemical presence, localized changes in turgor pressure altered the balance of expansion. Over time, these responsive tendencies became encoded within development itself. Leaves began to curl even in the absence of immediate contact, forming partial enclosures that increased the likelihood of retaining organic matter. At the same time, glandular cells multiplied and specialized. Instead of producing only protective mucilage, some began secreting a broader array of enzymes into the shallow basin formed by the curling blade. Proteases cleaved peptide bonds within proteins, reducing complex molecules into absorbable fragments. Phosphatases liberated phosphate groups from organic compounds. Chitinases weakened the structural polymers found in arthropod exoskeletons. These enzymes were not entirely new inventions. They arose from modifications of proteins already present in plant genomes, repurposed through gene duplication and altered regulation. As secretion increased, the leaf surface changed in texture. Cuticular layers thinned in localized regions to permit absorption. Transport proteins embedded within cell membranes facilitated the movement of amino acids and ammonium into vascular tissues. What had once been incidental contact became a controlled exchange. The shallow curl helped concentrate fluids, reducing dilution from rainfall and allowing enzymatic reactions to proceed more effectively. With each incremental refinement, structure and function intertwined more closely. Some leaves developed more pronounced folding, creating pockets that reduced escape. Others produced stronger secretions, capable of maintaining contact for extended periods. Developmental pathways shifted so that trap formation became a predictable part of leaf identity rather than a variable response. The transformation did not occur in a single lineage alone. In separate corners of nutrient-limited landscapes, similar modifications unfolded independently, guided by the same pressures of scarcity. Generations layered complexity upon curvature. Vascular strands extended deeper into modified regions, ensuring efficient distribution of absorbed nutrients. Sensory mechanisms, still primitive compared to later forms, allowed cells to respond to repeated stimulation with increased secretion. The leaf was no longer only an organ of photosynthesis. It had become a structure that integrated capture, digestion, and absorption within a single coordinated surface. Through gradual architectural change, the first true traps took shape. They were not yet swift or elaborate, but they represented a decisive shift in plant morphology. A leaf that once served solely as a collector of light had begun to function as a chamber for chemical breakdown. In the steady arithmetic of evolution, curved tissue and external enzymes marked a new phase, where form itself carried the imprint of nutritional necessity. At the edge of open wetlands, where the ground remains thin and the horizon feels wide, small rosettes press close to the surface, each leaf spread like a patient hand. From their blades rise slender filaments tipped with luminous droplets, beads of mucilage that catch the light and shimmer against the green. These plants belong to the genus Drosera, commonly known as sundews, and their brilliance is not pigment but secretion, a clear adhesive composed of polysaccharides and water. The droplets refract sunlight in a way that resembles nectar. To a passing insect, the leaf appears jeweled and inviting, a surface rich with moisture in an otherwise sparse landscape. When a gnat or small fly alights, its legs adhere instantly to the viscous film. The more it struggles, the more contact it makes. Each movement draws additional tentacles inward as surrounding filaments respond with gradual curvature. This motion unfolds over minutes to hours, driven by differential cell elongation and changes in turgor pressure along the length of each tentacle. As the prey becomes further entangled, the leaf itself may begin to participate. Some species of Drosera exhibit lamina movement, the blade curling gently around the captured body, increasing surface contact. Glandular cells along the tentacle heads shift from mucilage production to enzyme secretion. Proteases and phosphatases are released into the adhesive matrix, initiating the breakdown of soft tissues. Chitinases weaken the exoskeleton, allowing internal nutrients to diffuse outward. The process is deliberate rather than rapid. There is no sudden snap, only a slow consolidation of contact, as if the leaf were drawing a boundary around what it has secured. Within the moist microenvironment created by mucilage and curvature, digestion proceeds externally. Amino acids, ammonium, and soluble phosphates accumulate along the epidermal surface. Specialized transport proteins embedded in cell membranes facilitate their uptake, channeling these molecules into vascular strands that distribute them through the plant's body. Drosera species remain rooted in soil that offers little nourishment, yet their leaves function as both sensor and stomach. Mechanical stimulation from struggling prey enhances enzyme production, while chemical cues from nitrogen-rich compounds further regulate secretion. The plant integrates these signals without nerves or muscles, relying instead on electrochemical gradients and hormonal pathways that coordinate response across tissues. Across continents and climates, sundews have diversified into numerous forms, from diminutive temperate species to larger tropical varieties. Despite differences in size and habitat, the core strategy remains consistent: adhesive capture, gradual enclosure, enzymatic digestion, and direct absorption through the leaf surface. The energy required to produce mucilage and enzymes is offset by the nutrients gained, particularly in substrates where nitrogen and phosphorus are scarce. In these glistening rosettes, the transformation of a leaf into a hunting structure is complete yet restrained. Light still fuels their growth, but the surface that once gathered only photons now gathers prey. Within each sparkling droplet, chemistry and curvature converge, sustaining a lineage that thrives through patience and persistent adaptation. In a narrow stretch of coastal plain along the Carolinas, where longleaf pines stand over open savannas and the soil remains sandy and thin, a different kind of leaf took shape. Dionea muscipula, the Venus flytrap, grows low to the ground in habitats repeatedly shaped by fire and rainfall, landscapes bright with sun yet chronically poor in nitrogen. Within this setting of constraint, its leaves divided into two distinct lobes hinged along a central midrib, forming a structure unlike any other plant in its community. Each lobe bears along its inner surface a series of delicate trigger hairs, slender projections that rise from specialized epidermal cells. When an insect wanders across the open trap, contact with one of these hairs generates a rapid change in membrane potential. An action potential travels across the leaf tissue, a swift electrical signal propagated through ion flux and coordinated cellular depolarization. The plant does not possess nerves, yet the transmission resembles the electrical language found in animal systems, carried through living tissue with remarkable speed. A single touch is not enough. The trap remains open unless a second stimulation follows within a brief interval, ensuring that random debris or raindrops do not trigger unnecessary closure. When the threshold is met, cells along the outer surface of the lobes release stored tension. Water shifts between cellular compartments, altering turgor pressure and causing the two halves to snap shut in less than a second. The movement is powered not by muscle but by elastic energy accumulated within the curved geometry of the leaf. Initially, the closing is incomplete, leaving small gaps between marginal spines. If the captured insect continues to struggle, additional mechanical stimulation reinforces the signal. The trap seals more tightly, edges interlocking to form a temporary chamber. Within this enclosed space, digestive glands lining the inner surface begin to secrete a clear fluid, rich in proteases, phosphatases, and other hydrolytic enzymes. Secretion increases only after repeated confirmation of living prey, a regulatory safeguard that conserves energy in an environment where every resource is costly. Over several days, the insect's soft tissues are reduced to soluble compounds. Amino acids and ammonium ions diffuse across glandular surfaces and enter the vascular system, supplementing nutrients drawn from impoverished soil. Once digestion is complete, the trap reopens, revealing only the indigestible remnants of exoskeleton. The leaf can repeat this cycle a limited number of times before senescing, its investment carefully balanced against return. In Dionea muscipula, sensitivity, motion, and digestion converge within a single organ. The trap embodies an intricate coordination of electrical signaling and mechanical design, refined through generations under persistent nutrient limitation. Rooted in sand, yet capable of swift movement, the plant maintains its dependence on sunlight while integrating a rapid, predatory response. Within the quiet wetlands where it evolved, this snap trap stands as a precise solution shaped by scarcity and sustained by subtle physiological control. In certain wetlands and forest clearings, leaves no longer spread outward as blades or folded inward along hinges. Instead, they deepened, their tissues elongating and fusing into vertical tubes that rose from the ground like quiet vessels. These structures, known as pitchers, are modified leaves shaped into chambers, their margins rolled and sealed so that what was once a flat surface became an interior space. Light still falls upon them, yet their architecture directs attention downward toward a hollow core. Along the upper edge of each pitcher, the rim develops a specialized region called the peristome, a lip often ridged and coated with nectar. Glands along this margin secrete sweet fluids that attract insects searching for energy. The surface appears stable, yet it is engineered with precision. Microscopic grooves channel water across the rim, and when humidity rises or rain falls, the peristome becomes slick. An insect, stepping forward, loses traction and slides inward, unable to regain footing against the steep inner walls. The interior of the pitcher is lined with distinct zones. Near the top, a waxy layer reduces grip, causing prey to fall further. Lower down, downward pointing hairs discourage escape. At the base, digestive fluid collects, a mixture of water, enzymes, and dissolved compounds. Proteases and phosphatases work within this pooled solution, breaking down soft tissues into absorbable molecules. The chamber functions as a passive trap, relying on gravity and surface chemistry rather than rapid movement. In several species, digestion is not carried out by plant enzymes alone. Microbial communities inhabit the fluid, forming a miniature ecosystem within the pitcher. Bacteria contribute to decomposition, accelerating the breakdown of prey. In some cases, larvae of certain insects live within the fluid without being digested, feeding on captured material and in turn releasing waste products that the plant can absorb. These symbiotic relationships transform the pitcher into a contained habitat where nutrient cycling occurs on a small scale. Pitcher plants have arisen independently in different regions of the world. In North America, genera such as Sarracenia occupy acidic bogs and pine savannas. In Southeast Asia, Nepenthes species climb through tropical forests, their pitchers suspended from tendrils. Though separated by geography and evolutionary lineage, their solutions converge on a similar form, tubular leaves designed to collect and digest fallen prey. Convergent evolution shaped these chambers under parallel pressures of nutrient scarcity. Within each pitcher, architecture replaces motion. There is no snap, no curling blade, only a steady reliance on structure and fluid. In landscapes where soil offers little, these vertical leaves extend the boundary of nutrient acquisition above the ground. Prey drawn by nectar descends into a contained world of enzymes and microbes, and the plant absorbs what the soil cannot provide. Through patient design rather than speed, the pitfall engineer secures its share of scarce elements. Beneath the surface of still freshwater ponds and slow-moving streams, where stems drift freely without anchoring roots, delicate threads extend in quiet suspension. Among them, live members of the genus Utricularia, known as bladderworts, plants that have relinquished conventional roots and instead float or rest lightly within the water column. Their leaves are finely divided into branching segments, and along these segments, small, translucent bladders are attached, each no larger than a grain of sand. These bladders are not passive sacs. Their walls are elastic and slightly concave, held under tension by a subtle difference in pressure between interior and surrounding water. Glandular cells lining the inner surface actively pump ions outward, drawing water with them through osmosis. As fluid exits, the bladder compresses inward, creating a partial vacuum within. The trap is set not by motion but by preparation, a steady expenditure of energy that primes the chamber. At the entrance of each bladder sits a flexible door sealed by surface tension and supported by minute trigger hairs. When a small aquatic organism, such as a water flea or protozoan, brushes against these hairs, the mechanical disturbance destabilizes the door. It swings inward in a fraction of a second, and the pressure difference pulls surrounding water into the cavity. The prey is carried with it, drawn through the opening by the sudden inrush. The door then closes just as swiftly, restoring the sealed chamber. The entire sequence unfolds within milliseconds, among the fastest movements recorded in the plant kingdom. Yet the speed is silent and invisible to the unaided eye, occurring beneath the water's surface without ripple or disturbance. Once inside, the prey is confined within a fluid environment enriched with digestive enzymes. Proteases and other hydrolytic compounds begin breaking down soft tissues, releasing nitrogenous molecules and other nutrients. Absorptive cells lining the bladder wall transport these products into the plant's tissues, supplementing what little can be obtained from the surrounding water. Unlike terrestrial traps that rely on gravity or adhesive surfaces, bladderworts depend on rapid hydraulic mechanics. Their bladders reset after each capture, slowly pumping water outward again to reestablish the vacuum. This cyclical process repeats throughout the life of the plant, allowing numerous captures across a season. In nutrient-poor aquatic habitats, where dissolved nitrogen is often limited, this capacity offers a distinct advantage. The architecture of Utricularia demonstrates a different path toward carnivory, one shaped by buoyancy and fluid dynamics rather than by curvature or enclosure. Suspended in water, lacking substantial roots, these plants transform their leaves into micro-scale suction devices. Within each tiny bladder, pressure gradients and elastic walls cooperate to secure fleeting prey. Through precise control of ions and membranes, the plant converts physical tension into nutritional gain, extending the reach of photosynthetic life into the hidden currents of freshwater ecosystems. Within the sealed chambers and folded surfaces of these specialized leaves, a quiet chemistry unfolds, one that mirrors processes more commonly associated with animal bodies. The captured prey does not simply decay. It is methodically reduced through enzymatic action, its complex tissues dismantled into absorbable fragments. What appears from the outside as stillness conceals a coordinated cascade of reactions guided by proteins that have been subtly reshaped across evolutionary time. Proteases take the first role in this transformation. These enzymes target peptide bonds within proteins, cleaving long chains of amino acids into shorter segments and eventually into individual units. The structural framework of muscle fibers and cellular machinery begins to unravel as proteolysis proceeds. The released amino acids diffuse through the thin films of digestive fluid and approach the glandular surface of the leaf, where membrane transporters facilitate their entry into plant cells. Alongside proteases, phosphatases operate within the same confined space. Phosphorus, often bound within organic molecules such as nucleotides and phospholipids, becomes accessible only after specific bonds are broken. Phosphatases liberate phosphate groups, allowing this scarce element to enter metabolic pathways essential for energy transfer and nucleic acid synthesis. In nutrient-limited habitats, each liberated phosphate represents a measurable gain. Chitinases contribute to the dismantling of the insect exoskeleton. Chitin, a resilient polymer composed of repeating sugar derivatives, provides structural integrity to arthropod bodies. By hydrolyzing the glycosidic bonds within chitin, these enzymes weaken the outer casing, permitting internal tissues to be exposed to further digestion. The once protective shell softens, yielding its contents to enzymatic reduction. The fluid within many traps becomes mildly acidic during this process. Proton pumps embedded in glandular cells lower the pH, creating conditions that enhance enzymatic efficiency. Though not identical to the gastric environments of vertebrates, the chemistry converges in principle. Acidic surroundings facilitate protein denaturation, making substrates more accessible to proteases. The resemblance to stomach function arises not from shared ancestry with animals but from parallel solutions to the challenge of external digestion. At the genetic level, these capabilities did not emerge from entirely novel inventions. Gene duplication events provided additional copies of existing enzymes, freeing them from their original roles. Through gradual mutation and regulatory change, some copies became expressed in leaf glands rather than in roots or seeds. Promoters shifted, expression patterns altered, and proteins once involved in defense or internal recycling were redirected toward prey digestion. The transformation illustrates how evolution modifies available tools rather than creating entirely new ones. As digestion progresses, soluble nutrients cross the epidermal barrier and enter vascular tissues. The plant integrates these compounds into its metabolism, synthesizing chlorophyll, constructing new cells, and supporting reproductive structures. The trap then resets, awaiting another encounter. Within these leaves, digestion resembles that of animals in function, yet remains fully plant in origin, a convergence shaped by scarcity and refined through incremental genetic change. Across distant continents and separated lineages, similar solutions emerged in quiet repetition. In wetlands of North America, in tropical forests of Southeast Asia, in temperate heaths and remote plateaus, plants belonging to entirely different families began reshaping their leaves toward the same outcome. They shared no recent common ancestor that possessed traps, no singular origin point from which all carnivorous forms descended. Instead, their similarities arose independently, guided by parallel pressures that shaped form and function in comparable ways. Molecular phylogenetics has traced these lineages through chloroplast DNA and nuclear gene sequences, revealing that carnivory did not appear once and diversify outward. It arose at least 6 separate times in flowering plants. Sundews and Venus flytraps belong to one branch, pitcher plants in North America to another, tropical nepenthes to yet another, bladderworts to a lineage of their own. Even within these groups, variations unfolded independently, refining adhesive surfaces, snap mechanisms, pitfall chambers, and suction bladders under local constraints. This pattern reflects convergent evolution, the process by which unrelated organisms develop analogous structures when confronted with similar environmental challenges. Wings in birds and bats provide one example, enzymatic leaf traps provide another. The selective force was not predation itself, but nutrient limitation, particularly scarcity of accessible nitrogen and phosphorus in specific habitats. Where soil chemistry imposed consistent restriction, natural selection favored incremental traits that enhanced alternative nutrient acquisition. Each lineage began with distinct anatomical foundations. Some possessed glandular hairs predisposed toward secretion. Others had leaves capable of forming tubular shapes. Still others evolved finely divided aquatic structures that could be modified into bladders. Evolution did not erase their differences, it redirected them. Gene duplication events supplied raw material, regulatory networks, shifted expression patterns, and developmental pathways adjusted curvature, secretion, and sensitivity. Over time, these modifications coalesced into fully functional traps. The recurrence of carnivory across separate families suggests a powerful and persistent environmental signal. When similar ecological conditions appear, similar adaptive responses can follow, even among organisms with divergent ancestries. Evolution does not anticipate outcomes, yet it explores available variations continuously. When a particular configuration proves advantageous under given constraints, it can emerge again through different genetic routes. In each independent origin, photosynthesis remained central. Carnivory supplemented rather than replaced the capture of light. The repeated emergence of traps demonstrates that the plant body is more flexible than once assumed, capable of reorganizing its leaves into structures that digest and absorb animal tissue without abandoning its foundational metabolism. Through multiple origins, the narrative of carnivory becomes less an anomaly and more a recurring theme. Under sustained nutrient stress, separate evolutionary paths converged on comparable designs. The result is a scattered yet coherent pattern across the plant kingdom, where similar pressures shaped similar innovations in quiet parallel, reaffirming that evolution often revisits solutions when circumstances align. Every adaptation carries a price, even those that appear elegantly suited to their surroundings. The leaves that fold, seal, secrete, or draw inward are not free structures. They require carbon skeletons assembled through photosynthesis, proteins synthesized from limited nitrogen, membranes maintained across shifting conditions of moisture and light. To build a trap is to redirect resources that might otherwise extend roots or enlarge foliage. In habitats defined by scarcity, the investment must be carefully balanced against return. Carnivorous plants often grow with measured restraint. Their stems remain modest, their leaves specialized rather than expansive. Energy allocated to glandular tissue, trigger hairs, digestive enzymes, or hydraulic mechanisms cannot simultaneously fuel rapid vertical growth. The production of proteases, phosphatases, and other enzymes demands amino acids that are themselves costly to assemble. Even resetting a suction bladder or reopening a snap trap requires metabolic expenditure, drawing upon stored carbohydrates formed under sunlight. Photosynthesis remains the primary engine of survival. Chloroplasts continue to harvest photons, converting carbon dioxide and water into sugars that sustain respiration and growth. Carnivory does not replace this foundation. Instead, it supplements it, providing access to nitrogen, phosphorus, and trace elements that the soil withholds. The captured prey contributes nutrients that enhance protein synthesis and reproductive capacity, yet the energy to power digestion and trap construction originates from light. In environments where minerals are abundant and microbial communities efficiently recycle nutrients, the balance shifts. Plants without traps can allocate more carbon toward leaf area, stem height, and rapid seed production. They spread quickly, overshadowing slower growing neighbors. Carnivorous species, burdened by the structural and metabolic costs of their specialized leaves, often lose this competition. Their traps confer advantage only under persistent nutrient limitation. When the constraint is lifted, the adaptation becomes less favorable. This dynamic underscores a central principle of evolution. Traits persist not because they are inherently superior, but because they align with specific ecological contexts. The same mechanism that grants survival in a bog or sandy savanna may hinder performance in fertile meadow or forest loam. Trade-offs are woven into every physiological system, Investment in one function reduces availability for another, shaping form according to circumstance. Carnivorous plants embody this equilibrium. They remain rooted in sunlight, dependent on the same photosynthetic chemistry as their non-carnivorous relatives. Their traps extend the range of nutrient acquisition but demand maintenance and construction costs that cannot be ignored. Growth proceeds slowly, yet steadily, aligned with environments where speed is less important than persistence. Within the arithmetic of energy and matter, hunting becomes a calculated supplement, effective only where soil fails to provide. In certain tropical forests, where humidity hangs in the air and climbing vines thread through layered canopies, some pitchers grow larger than a human hand. Their chambers deepen and widen, supported by tendrils that suspend them above the forest floor. Within these expanded vessels, the scale of capture occasionally extends beyond the small arthropods that constitute the usual diet of carnivorous plants. Ants, flies, and beetles remain the most frequent visitors, yet in rare instances, frogs, lizards, and even small mammals have been found within the fluid at the base of these leaves. The majority of prey across carnivorous species consists of insects and other small invertebrates. Their abundance, mobility, and size align with the mechanical limits of most traps. Sticky surfaces immobilize flies, snap mechanisms enclose beetles, suction bladders draw in microscopic crustaceans. These interactions dominate the nutritional intake of carnivorous plants and form the steady background of their feeding ecology. Field measurements confirm that nitrogen derived from arthropods can significantly enhance chlorophyll content, leaf growth, and reproductive output in nutrient-poor habitats. Yet, among certain large Nepenthes species in Southeast Asia, observations have documented more substantial captures. A frog seeking shelter may slip along a slick peristome. A small rodent, drawn by nectar or scent, may lose footing and fall inward. In these uncommon events, digestion proceeds over extended periods as proteolytic and phosphatase activity reduces tissues into soluble compounds. The nutrient gain from a single vertebrate can exceed that obtained from dozens of insects, contributing measurable increases in foliar nitrogen concentration and seed production. Field studies have quantified these contributions through isotopic analysis, tracing nitrogen signatures from prey into plant tissues. In habitats where vertebrate capture occurs, the additional nutrients can represent a meaningful fraction of total nitrogen uptake. However, such cases remain exceptional rather than typical. The architecture of most traps is not optimized for large prey, and the energetic costs associated with maintaining extensive chambers or fluid volumes impose limits on frequency. These rare captures expand the ecological range of carnivory without redefining its core pattern. Small arthropods continue to form the principal nutritional source, aligning with trap dimensions and encounter rates. Larger prey, when present, represent opportunistic extensions of a strategy shaped primarily around invertebrate abundance. Through these occasional events, the boundary between plant and animal interaction broadens slightly, yet remains grounded in ecological reality. The digestion of a frog or rodent does not transform the plant into a dominant predator. It reflects the same principle that guided earlier adaptations, the capacity to utilize available nutrients when environmental scarcity persists. In shaded forests and open wetlands alike, carnivorous plants remain anchored in photosynthesis, supplementing their intake through whatever prey chance and structure allow. Within the fluid-filled chambers of certain pitchers, life does not end with capture. Instead, a contained community begins to assemble, turning the interior of the leaf into a small aquatic habitat. Rainwater mingles with digestive secretions, forming a pool that reflects the sky above while sheltering organisms below. In these enclosed spaces, the boundary between predator and host softens into something more intricate. Some species of pitcher plants provide refuge for mosquito larvae that develop entirely within the trap. The larvae do not succumb to digestion. Instead, they feed upon the remains of captured prey, shredding larger fragments into smaller particles. Their movement stirs the fluid, redistributing nutrients and oxygen within the confined chamber. In turn, the plant absorbs dissolved nitrogenous compounds released through larval waste and microbial processing. The relationship is not incidental but stable, maintained across generations in habitats where both partners benefit. Bacteria and protozoa contribute to this internal ecology as well. Microbial communities colonize the fluid, forming biofilms along the inner walls. They secrete enzymes that complement those produced by the plant, accelerating the breakdown of proteins and other organic molecules. Through mineralization, complex tissues are converted into ammonium and phosphate, increasing the availability of nutrients for absorption. The plant provides structure and moisture. The microbes provide metabolic versatility. In some tropical pitchers, additional invertebrates inhabit the chamber, including mites and small crustaceans adapted to the fluid environment. These organisms feed on decomposing material or on one another, creating a miniature food web contained within a single leaf. Each participant occupies a niche, contributing to the gradual transformation of prey into soluble compounds. The trap becomes less a simple digestive vessel and more a micro-ecosystem sustained by continuous interaction. These symbiotic partnerships illustrate that carnivory need not operate in isolation. The plant sets the physical framework, shaping a chamber that collects water and prey, yet the subsequent processing may involve multiple species. Mutualistic relationships evolve when shared benefits outweigh competition. The mosquito larvae gain protection from external predators. The plant gains enhanced nutrient release. Microbes secure a stable habitat. The plant receives efficient decomposition. Such internal ecosystems remain bounded by the lifespan of the leaf. When the trap senesces, its contained community disperses or perishes. Yet during its active phase, the pitcher supports a network of exchanges that mirrors larger ecological systems in compressed form. Nutrients flow from captured organisms through microbial intermediaries into plant tissue, while inhabitants depend on the plant's architecture for survival. Within these chambers, carnivory intertwines with cooperation. The leaf becomes both hunter and host, sustaining a contained web of life that transforms scarcity into shared persistence. Long before any living trap closed around an insect, subtle traces were already being pressed into stone. In sediment layers laid down beneath ancient wetlands and shallow lakes, pollen grains settled and hardened, preserving microscopic signatures of lineages that no longer exist in their original form. Under magnification, the sculpted walls of these grains reveal affinities with modern carnivorous families, linking present adaptations to deep evolutionary roots. Fossilized leaves provide rarer but more direct clues. Impressions of tubular structures, curved margins, and glandular surfaces appear in certain deposits, suggesting that modified foliage, capable of capture, had already taken shape millions of years ago. These remnants are often fragmentary, compressed by time and tectonic pressure, yet their outlines echo the architectures seen in living species. Though soft tissues seldom endure, the persistent shapes of veins and margins offer evidence that traps are not recent innovations. Beyond visible fossils, molecular clock analyses extend the timeline further. By comparing rates of genetic mutation across related species, researchers estimate when carnivorous lineages diverged from their non carnivorous relatives. These calculations place several origins during the Eocene epoch, a period characterized by warm global climates and widespread wetland habitats. While precise dates remain refined with each new dataset, the convergence of genetic and paleobotanical evidence situates the rise of carnivory within ancient environmental transitions. The Eocene landscape differed markedly from the present. Elevated temperatures and high atmospheric carbon dioxide supported expansive forests and humid lowlands. In many regions, rainfall patterns and geological processes created nutrient poor substrates similar to those where carnivorous plants thrive today. Peat accumulating basins, acidic soils, and sandy deposits would have imposed the same elemental limitations that shaped later adaptations. Within such settings, incremental modifications of leaf form and enzyme production could be preserved through natural selection. Fossil pollen attributed to families such as Droseraceae and related groups appears in strata dating back tens of millions of years. These grains, resistant to decay due to their sporopollenin walls, survive long after leaves and stems vanish. Their distribution across ancient sediments indicates that carnivorous or proto-carnivorous ancestors were already diversifying across continents during warm climatic intervals. The alignment of fossil structures, pollen records, and molecular estimates forms a coherent narrative. Carnivory did not arise abruptly in recent history. It unfolded gradually within specific climatic and ecological contexts. As global temperatures shifted and habitats fragmented, lineages bearing early trapping traits persisted where nutrient stress remained constant. Through fragments of leaf impressions and microscopic pollen grains, the deep antiquity of these adaptations becomes visible. Stone retains the faint geometry of ancient traps, and DNA preserves the tempo of divergence. Together, they reveal that the capacity to digest prey has endured across epochs, shaped by climates long vanished yet echoed in modern wetlands where carnivorous plants still persist. Beneath the still surface of a leaf, currents move without sound. When a trigger hair is bent or a surface is disturbed, a shift begins at the level of individual cells. Membranes alter their permeability, ions flow across channels, and an electrical impulse propagates through living tissue. In species such as the Venus flytrap, this impulse resembles an action potential, a rapid change in voltage that travels from one region of the leaf to another, coordinating movement and secretion. The signal begins when mechanosensitive channels open in response to touch. Sodium, potassium, and other ions move according to electrochemical gradients, creating a transient depolarization of the membrane. This disturbance does not remain local. Through plasmodesmata and vascular strands, the electrical change spreads, linking distant cells in a shared response. Within moments, the leaf receives the message that contact has occurred. Calcium ions play a central role in translating this electrical event into biochemical action. As the membrane potential shifts, calcium channels open, allowing an influx into the cytoplasm. Elevated calcium concentrations act as second messengers, binding to proteins that regulate gene expression and enzyme secretion. In carnivorous species, repeated stimulation amplifies this cascade, ensuring that digestive glands activate only after consistent mechanical cues. The plant integrates electrical and chemical information, filtering noise from meaningful contact. Unlike animal neurons, plant cells lack specialized nerve fibers insulated by myelin or organized into centralized networks. There is no brain coordinating decisions. Instead, electrical signals move through ordinary tissues, including phloem and parenchyma, using ion channels embedded in membranes common to all plant cells. The speed of transmission is slower than in vertebrate nerves, yet sufficient to coordinate rapid movements such as trap closure. These signals do not serve carnivory alone. Electrical communication in plants also responds to wounding, temperature shifts, and changes in light. A damaged leaf can send a voltage signal to neighboring tissues, prompting the production of defensive compounds. A sudden environmental stress can trigger systemic adjustments in stomatal conductance or growth patterns. The capacity for internal communication is widespread, though most visible in species whose movements reveal its presence. In carnivorous plants, electrical signaling becomes part of a finely tuned sequence. Mechanical stimulation generates action potentials. Calcium waves translate them into metabolic shifts. Enzymes are secreted. Traps close or tighten. Each stage relies on coordinated ion movement and membrane dynamics. The absence of neurons does not prevent complex response. It redirects it through alternative cellular architecture. Within these leaves, communication unfolds without synapses or centralized control. Electrical gradients rise and fall, linking cells across a surface that once seemed passive. The distinction between plant and animal signaling becomes less rigid when examined at the level of ions and membranes. Through shared physical principles and divergent anatomical paths, plants have evolved their own language of electricity, quiet yet precise, guiding movements and digestion within tissues anchored firmly in the soil. Language often moves faster than biology. When a leaf closes or a chamber fills with prey, the word hunting rises easily to the surface of description. Yet within scientific circles, that word carries weight, shaped by definitions developed in the study of animals. Hunting traditionally implies pursuit, sensory tracking, and intentional movement across space. Carnivorous plants complicate that framework, existing at the edge of categories that once seemed clear. Some traps remain entirely passive in their operation. Pitchers rely on gravity and surface structure. Adhesive leaves depend on contact and retention. Suction bladders prime themselves and wait. Even the rapid snap of a flytrap is confined to the space of a single leaf. There is no roaming across the landscape, no directed chase. The plant remains rooted, its architecture positioned within an environment where prey arrives through chance and attraction rather than pursuit. From an evolutionary perspective, predation can be defined more broadly as the consumption of one organism by another to obtain nutrients. By this definition, carnivorous plants qualify without ambiguity. They capture, digest, and absorb tissues from living organisms. Their enzymes dismantle proteins and structural polymers, integrating the resulting molecules into their own metabolism. The absence of locomotion does not negate the transfer of energy and matter from prey to predator. Ecologically, their role remains modest. Most carnivorous species occupy specialized niches within nutrient-limited habitats. They do not regulate prey populations on a broad scale, nor do they reshape food webs in the manner of large animal predators. Instead, they operate at a small scale, removing insects that happen to encounter their traps. Their impact is localized, intertwined with the dynamics of wetland and heath ecosystems where invertebrates are abundant. The debate over terminology reflects deeper questions about agency and behavior. Plants respond to stimuli through biochemical and electrical pathways, yet their responses are bound to fixed positions. The term hunting evokes imagery of movement and intention, qualities that plants express differently, if at all. Some researchers prefer to describe these interactions as carnivory or nutrient acquisition rather than predation, emphasizing process over metaphor. Still, the distinction is largely conceptual. Whether labeled hunting or passive capture, the functional outcome remains the same. Organic matter is secured, broken down, and assimilated. The plant benefits in environments where soil nutrients are scarce. Evolution does not concern itself with vocabulary. It favors mechanisms that improve survival and reproduction within given constraints. In examining these leaves and chambers, the language chosen shapes perception, but not biology. Carnivorous plants stand at an intersection between categories, absorbing elements of both plant and predator. Their existence invites careful use of words, acknowledging both the limits of traditional definitions and the fluidity of life's strategies. Across the vast diversity of flowering plants, carnivory occupies only a narrow margin. Out of more than 390,000 described plant species worldwide, roughly 800 are recognized as truly carnivorous. The proportion is small, almost a statistical whisper within the greater fabric of terrestrial life. Forest canopies, grasslands, deserts, and alpine slopes are dominated by plants that rely entirely on soil nutrients and sunlight, leaving specialized traps confined to select ecological pockets. This rarity reflects the demanding conditions required for carnivory to offer an advantage. The habitats that favor it are not common. Nutrient-poor wetlands, acidic seeps, sandy savannas, and oligotrophic waters form scattered mosaics rather than continuous expanses. Within these environments, nitrogen and phosphorus remain persistently limited, creating the narrow window where trapping prey can offset energetic costs. Outside of such settings, conventional strategies prevail. Energetic constraints shape this distribution. Constructing adhesive glands, hydraulic bladders, hinged lobes, or deep tubular chambers requires carbon investment derived from photosynthesis. Producing digestive enzymes demands amino acids that must themselves be assembled from scarce nitrogen. Maintaining sensitivity to mechanical or chemical cues involves metabolic regulation and ion transport. These processes consume resources that could otherwise support rapid growth or competitive expansion. In nutrient-rich soils, plants that allocate energy toward expansive leaves and tall stems often outcompete slower-growing carnivorous neighbors. They capture more light, disperse more seeds, and establish dense populations that shade smaller species. Under such circumstances, the cost of maintaining traps outweighs the benefit. Selection then favors forms that channel energy directly into growth rather than into capture mechanisms that are no longer necessary. Even within suitable habitats, not every lineage evolves carnivory. The transition requires specific pre-existing traits, such as glandular surfaces capable of secretion or leaf architectures amenable to modification. Genetic pathways must be available for duplication and repurposing. Environmental pressure alone does not guarantee the emergence of traps. It interacts with historical contingency and developmental potential. The scattered presence of carnivorous plants across continents underscores this interplay. They appear where ecological constraints persist and where ancestral structures allowed adaptation. Yet they remain absent from most ecosystems, not because the strategy fails, but because the balance of costs and benefits rarely aligns in its favor. Carnivory endures as a specialized response rather than a dominant mode of life. It thrives in edges and margins, in soils washed thin by rain or saturated beyond aeration. Within those niches, the arithmetic of energy and nutrients permits its persistence. Beyond them, the broader plant kingdom continues along pathways shaped by more conventional exchanges between root, soil, and sun. Climate has never remained still for long. Across geological time, temperature curves have risen and fallen, ice sheets have advanced and retreated, and rainfall patterns have reorganized entire landscapes. During intervals of global warming, wetlands expanded into new latitudes, sea levels shifted, and previously temperate regions grew humid. These changes did not create carnivory, but they shaped the spaces in which it could persist and diversify. In warmer periods of the past, elevated atmospheric carbon dioxide and increased precipitation fostered broad lowland wetlands and peat forming basins. Such environments provided the nutrient poor, water saturated conditions that favor trapping adaptations. As climates shifted, these habitats appeared, fragmented, and reformed in different configurations across continents. Carnivorous lineages that were already adapted to scarcity found new territories where their strategies remained advantageous. Habitat shifts also imposed geographic isolation. When warming trends were followed by cooling or drying, wetlands contracted into refugia, separated by stretches of unsuitable terrain. Populations once connected became divided. Over time, genetic divergence accumulated within these isolated groups. Small differences in leaf structure, secretion rate, or growth habit were preserved through local selection, eventually producing distinct species. Speciation in carnivorous plants often mirrors the patchwork nature of their habitats, each lineage tied to specific hydrological and climatic conditions. In tropical regions, climatic oscillations influence the distribution of montane and lowland forests, altering light availability and moisture regimes. Climbing pitcher plants extended into new niches as forest canopies opened or shifted. In temperate zones, cycles of glaciation and interglacial warming repeatedly reshaped wetlands, pushing populations southward during cold intervals and allowing recolonization during warmer phases. Each transition filtered genetic variation through the lens of survival in nutrient-limited substrates. Climate change did not uniformly benefit carnivorous plants. Rapid environmental alterations could eliminate specialized habitats as easily as create them. When wetlands drained or were replaced by richer soils, species dependent on scarcity faced contraction. The spread of carnivory across landscapes has therefore been episodic rather than continuous, tracking the rise and fall of specific ecological conditions. Through these shifting climates, carnivorous plants demonstrated resilience rooted in specialization. Their ability to capitalize on nutrient-poor environments allowed them to persist when competitors faltered. Yet their distribution remained closely tied to hydrology, temperature, and soil chemistry, variables shaped by global atmospheric patterns. The history of their spread is intertwined with Earth's changing climate. Warming events opened corridors of opportunity. Cooling phases imposed isolation. Both processes contributed to diversification. Within the broader narrative of planetary change, carnivorous plants moved quietly across continents, their traps evolving not in isolation from climate, but in dialogue with it. If one were to trace their presence across a map of the modern world, carnivorous plants would appear not as a continuous band, but as scattered constellations. They are found on every continent except Antarctica, yet their distribution is uneven, clustered in regions where soil chemistry and water regimes align with their specialized needs. From the pine savannas of the southeastern United States to the peat bogs of northern Europe, from the tropical forests of Southeast Asia to isolated plateaus in Australia and South America, their habitats form a patchwork tied closely to hydrology and climate. Wetlands remain their primary stronghold. Acidic bogs, nutrient poor fens, sandy coastal plains, and seasonally waterlogged grasslands provide the persistent scarcity that favors trapping adaptations. In tropical zones, high rainfall and leached soils create similar conditions beneath dense canopies or along open clearings. There, climbing pitcher plants suspend their traps from tendrils, while ground dwelling species anchor themselves in thin substrates. In temperate regions, sundews and butterworts occupy sphagnum moss carpets and seepage slopes where mineral input remains limited. Geographic isolation shapes this distribution. Many carnivorous species are restricted to narrow ranges, sometimes confined to a single watershed or mountain range. Such confinement reflects both habitat specificity and limited dispersal. Seeds may travel by wind or water, yet successful establishment requires precise combinations of moisture, acidity, and nutrient scarcity. Where these conditions fail to converge, populations do not persist. On continents such as Australia and South America, ancient geological stability and diverse microhabitats have fostered notable radiations. Bladderworts inhabit freshwater systems across broad latitudinal gradients, while terrestrial species occupy ephemeral pools and damp sands. In Southeast Asia, complex topography and climatic gradients have encouraged diversification among pitcher plants, with distinct species adapted to lowland forests, montane slopes, or limestone outcrops. Each region supports forms shaped by local conditions, reflecting evolutionary pathways influenced by both isolation and environmental filtering. Despite their wide geographic reach, carnivorous plants remain absent from vast stretches of fertile plains, dense agricultural soils, and arid deserts. Their presence signals not abundance of nutrients, but their absence. Where ecosystems provide ample nitrogen and phosphorus, conventional plant strategies dominate, leaving traps unnecessary and energetically disadvantageous. The global distribution of carnivory, therefore, mirrors a map of constraint. It outlines the margins where water saturates the ground, yet minerals remain scarce, where rainfall strips soils of fertility, and where ecological niches persist in delicate balance. Across continents, these plants occupy scattered refuges shaped by geology and climate, each population a quiet testament to adaptation within narrow ecological corridors. Within the quiet spaces where traps wait, another story unfolds among the insects that move through them. Prey are not passive participants in this exchange. Over generations, populations exposed to carnivorous plants experience selective pressure of their own. Individuals that avoid capture leave more offspring, and subtle behavioral or structural differences accumulate within their lineages. In habitats dense with adhesive leaves or tubular chambers, certain insects alter their approach patterns. Some species learn to feed cautiously along the margins of traps, harvesting nectar without stepping fully onto perilous surfaces. Others reduce the time spent on slippery rims or shift activity to hours when traps are less effective. Behavioral plasticity, even modest in scale, can influence survival rates across many generations. Morphological changes may also arise. Stronger legs, altered tarsal structures, or variations in body size can affect the likelihood of slipping into a chamber or becoming immobilized by mucilage. In aquatic environments, microcrustaceans capable of sensing pressure fluctuations may evade suction bladders more effectively than less sensitive counterparts. Sensory acuity, speed of response, and flight patterns all contribute to the delicate balance between capture and escape. In response, plants are not static. Selection continues to refine trap efficiency. In some pitcher plants, the microstructure of the peristome enhances slipperiness under specific humidity conditions, reducing the chance of prey gaining traction. Adhesive secretions may vary in viscosity or in the spectrum of volatile compounds that attract insects. Snap traps may fine-tune trigger hair sensitivity, minimizing false closures while maintaining rapid response to genuine prey. Even the composition of digestive fluids can shift, optimizing breakdown of the most commonly captured organisms. This reciprocal influence resembles an ecological arms race, though it unfolds slowly and without intention. Each adaptation in prey exerts pressure on plant populations, favoring modifications that restore capture success. In turn, improved trapping mechanisms select for further avoidance traits in insects. The cycle is not symmetrical in scale, yet it persists as long as both partners occupy the same habitat. Such interactions contribute to diversification. Local insect communities differ from region to region, and plant populations adapt to the specific assemblages they encounter. A species of carnivorous plant in one wetland may emphasize attraction through scent, while another in a different environment relies more heavily on structural design. Insects likewise evolve within the context of their local predators, shaping behaviors that reflect particular risks. Through this ongoing exchange, carnivory becomes embedded within broader ecological networks. Traps and wings, secretions and exoskeletons all adjust incrementally under mutual influence. The result is not domination by one side, but a dynamic equilibrium shaped by countless encounters, each leaving a faint imprint on the next generation. Before any trap closes or chamber fills, an invitation is often extended in forms too subtle for the human eye alone. Along the rims of pitchers, across the surfaces of adhesive leaves, and near the entrances of concealed bladders, chemical signals drift outward. These signals are not random byproducts. They are shaped through selection to influence the sensory worlds of insects that move through wetland air and forest understory. Nectar production is one of the most direct lures. Glands positioned near perilous surfaces secrete droplets rich in sugars and amino acids. For an insect navigating landscapes where energy must be gathered continuously, such exudates represent valuable resources. The sweetness encourages exploration, drawing bodies closer to slippery edges or sticky tentacles. In some species, nectar flow increases during periods of active growth, aligning attraction with the plant's capacity to digest and absorb nutrients. Color also plays a role beyond what human vision detects. Many carnivorous plants exhibit patterns visible in the ultraviolet spectrum, a range of light perceptible to numerous insects. Contrasting patches and guiding lines form visual cues that lead toward trap openings. Under ultraviolet illumination, what appears uniformly green may reveal striking signals, directing pollinators and prey along specific pathways. These patterns can function like landing guides, focusing movement toward zones where capture probability rises. Scent adds another layer to this chemical dialogue. Volatile organic compounds released from glands can mimic odors associated with food sources or decaying matter. Certain pitcher plants emit fragrances reminiscent of ripe fruit, while others produce subtler blends that resemble nectar-bearing flowers. Insects respond to these cues based on evolved sensory biases, approaching sources that promise nourishment. The plant exploits these responses without movement, relying on diffusion and air currents to carry its message. This chemistry of attraction is finely balanced. Excessive lure could overwhelm digestive capacity, while insufficient signaling would reduce encounter rates. Some species even vary their volatile profiles in response to environmental conditions, adjusting emission rates according to humidity or temperature. The interplay between secretion, light reflection, and scent production forms an integrated strategy that precedes mechanical capture. Within this system, attraction is not deception in a moral sense, but an ecological interaction shaped by mutual constraints. Insects seek energy, plants seek nutrients unavailable in soil. The exchange begins with sensory engagement, guided by sugars, ultraviolet contrasts, and airborne molecules that bridge the space between root and wing. Through nectar droplets, spectral patterns, and aromatic plumes, carnivorous plants extend their reach beyond static structures. They engage the perceptual systems of other organisms, inviting contact that may end in assimilation. In these quiet wetlands and forests, chemistry becomes language, and attraction becomes the first step in a process that sustains life under conditions of persistent scarcity. In laboratories far removed from wetlands and forest clearings, another layer of the story has been unfolding within strands of DNA. Advances in genome sequencing have allowed researchers to read the full genetic blueprint of species, such as the Venus flytrap, revealing how carnivory is encoded at the molecular level. What emerges from these analyses is not an entirely novel genetic architecture, but a reshaped version of tools long present in plant lineages. The genome of the Venus flytrap shows clear shared ancestry with non-carnivorous relatives within the same broader family. Its chromosomes contain genes common to many flowering plants, including those involved in photosynthesis, growth regulation, and stress response. Carnivory did not arise from a separate lineage detached from conventional plant biology. Instead, it evolved within an existing framework, modifying and amplifying pathways that once served different purposes. Gene duplication plays a central role in this transformation. When a gene is copied during replication, one version can maintain its original function while the other becomes free to accumulate mutations. Over time, duplicated genes may diverge in expression pattern or enzymatic specificity. In carnivorous plants, genes originally associated with defense, wound response, or nutrient recycling have been co-opted for digestive roles. Proteases that once helped dismantle internal proteins during senescence now act externally upon captured prey. Transporters that once moved nutrients within the plant now facilitate absorption from trap surfaces. Sequencing data also reveal regulatory changes. Promoter regions upstream of key genes have evolved to activate expression in trap tissues rather than in roots or leaves more broadly. Electrical stimulation of trigger hairs leads to shifts in gene expression within digestive glands, linking sensory input to metabolic output through networks of transcription factors. Calcium-dependent signaling cascades intersect with these genetic controls, coordinating enzyme production with confirmed prey capture. Comparative genomics across multiple carnivorous species underscores a recurring pattern. Different lineages have modified overlapping sets of genes, even when their traps differ structurally. This suggests that certain molecular pathways are particularly amenable to repurposing under nutrient limitation. Evolution repeatedly draws upon similar genetic reservoirs, reshaping them through duplication and regulatory adjustment rather than inventing entirely new sequences. The modern genetic perspective reframes carnivory as an example of innovation through modification. Beneath the visible architecture of traps lies a genome that bears traces of ordinary plant ancestry, interwoven with specialized adaptations. Sequencing has illuminated how incremental changes at the level of DNA translate into complex physiological traits. Within each carnivorous plant, the memory of its lineage remains written in nucleotides. Genes shared with non-carnivorous relatives coexist alongside duplicated and redirected counterparts. The transformation from passive leaf to active trap is inscribed not in a single dramatic mutation, but in layers of genetic refinement accumulated across deep time. In the quiet wetlands and sandy seeps where carnivorous plants take root, nutrients do not move in abundance. They circulate slowly, constrained by water saturation, acidity, and thin mineral reserves. Within these restrained systems, every pathway of nitrogen and phosphorus matters. Carnivorous plants insert themselves into this circulation not as dominant forces, but as subtle redirectors of flow. When an insect becomes prey, its body shifts from mobile participant in the ecosystem to localized resource. Nitrogen bound within muscle tissue, phosphorus stored in nucleic acids, and trace elements carried within hemolymph are drawn into plant tissue rather than returned directly to the soil through decomposition. The plant absorbs these compounds, incorporating them into chlorophyll, enzymes, and reproductive structures. Through this transfer, nutrients that once moved horizontally through insect life cycles become anchored vertically within leaf and stem. This redirection influences local nutrient cycling in small but measurable ways. Studies using stable isotope analysis reveal that a portion of the nitrogen in carnivorous plant tissues originates from captured prey. When leaves senesce and fall, these nutrients eventually re-enter the surrounding substrate, enriching microsites within otherwise impoverished soils. In this manner, carnivory can create localized patches of slightly higher fertility, influencing the growth of neighboring mosses or seedlings. The impact on insect populations is generally modest. Most carnivorous plants capture small arthropods that are abundant within their habitats. The removal of individuals rarely alters overall population dynamics at broad scales. However, at the microhabitat level, traps can affect species composition, favoring insects less susceptible to capture while reducing the abundance of those frequently ensnared. These subtle shifts ripple outward through predator and competitor relationships, integrating carnivorous plants into wider ecological networks. In some pitcher plant communities, the internal ecosystems discussed earlier further complicate nutrient pathways. Microbes and invertebrates living within traps process prey remains, releasing mineralized compounds that the plant absorbs. Waste products from these organisms contribute additional nitrogen to the system. When fluid overflows during heavy rain, dissolved nutrients may enter surrounding moss, mats, or soil, extending the influence of a single trap beyond its immediate structure. Carnivory does not overturn ecosystem function. Photosynthesis remains the primary driver of energy input, and decomposition continues to recycle organic matter broadly. Yet in habitats where nutrient input is chronically limited, the capture of insects provides an auxiliary channel of redistribution. It links aerial insect biomass to rooted plant tissues, bridging two compartments of the ecosystem that would otherwise exchange resources more slowly. Through these incremental transfers, carnivorous plants participate in the quiet choreography of nutrient cycling. They do not dominate the flow, but they adjust it, capturing fleeting elements from moving bodies and weaving them into stationary forms. In landscapes defined by scarcity, even such modest redirection shapes the persistence of life. Across many of the fragile landscapes that sustain carnivorous plants, change now arrives with unusual speed. Wetlands are drained for agriculture and development, peatlands are mined for fuel and horticulture, and sandy savannas are converted into plantations or paved surfaces. The thin soils and waterlogged basins that once provided the specific conditions required for trapping species are altered or removed entirely. When hydrology shifts even slightly, the balance of acidity and nutrient availability can change, rendering habitats unsuitable within a single human generation. Wetland loss poses a particular threat. Many carnivorous plants depend on consistent water tables and seasonal flooding patterns. Drainage ditches lower groundwater levels, exposing peat to oxidation and increasing nutrient input from surrounding land. As soils become richer in nitrogen and phosphorus, faster growing competitors establish themselves, shading and displacing specialized species that evolved under scarcity. The same adaptation that once ensured survival becomes a disadvantage in newly enriched environments. Illegal plant trade compounds these pressures. The striking appearance of certain species, especially large pitcher plants and Venus flytraps, has fueled demand in horticultural markets. Although many are cultivated legally, wild populations are sometimes harvested unsustainably. Removal of mature individuals reduces reproductive output and genetic diversity, particularly in species already restricted to small geographic ranges. In isolated habitats, even modest extraction can have long-lasting effects. Climate sensitivity further complicates conservation. Carnivorous plants often occupy narrow ecological niches defined by temperature, precipitation, and soil chemistry. As global climates shift, suitable habitats may move poleward or upward in elevation. Yet dispersal to newly favorable areas is not guaranteed. Fragmented landscapes and human-altered terrain can block migration corridors, leaving populations stranded in environments that no longer match their physiological tolerances. Increased drought frequency or altered rainfall timing can disrupt the hydrological regimes essential for survival. Conservation efforts increasingly focus on habitat protection and restoration, preserving intact wetlands, regulating water extraction, and preventing nutrient runoff help maintain the low fertility conditions these species require. Cultivation programs aim to reduce pressure on wild populations by supplying legally propagated plants to collectors. In some regions, reintroduction projects seek to restore species to sites where they have declined. The vulnerability of carnivorous plants underscores the delicacy of the ecosystems they inhabit. Their persistence depends not only on their unique adaptations, but on the stability of water tables, soil chemistry, and climatic patterns. Within landscapes already shaped by scarcity, additional stress can tip the balance. Safeguarding these species, therefore, involves protecting the subtle environmental parameters that allowed their evolution in the first place. When examined across deep time, carnivorous plants reveal less about predation than about constraint. Their existence is a record of adaptation under persistent stress, where scarcity did not eliminate lineages but reshaped them. In soils stripped of accessible nitrogen and phosphorus, ordinary strategies proved insufficient. Yet rather than abandoning those environments, certain plants modified themselves incrementally, preserving photosynthesis while extending their reach into new nutrient sources. This pattern reflects a broader principle of evolution. Novelty rarely appears as sudden invention. It arises through alteration of existing structures, redirected by selection toward new functions. Leaves that once served only to harvest light were reshaped into chambers, adhesive fields, or hinged surfaces. Enzymes that originally processed internal proteins or defended against pathogens were redeployed to digest external prey. Electrical signals that coordinated wound responses became integrated into rapid trap movements. Each innovation emerged from materials already present within the plant body. Such transformations illustrate how biological systems carry latent potential. Gene duplication creates redundancy. Regulatory shifts change where and when proteins are expressed. Structural variation alters mechanical properties. Under sustained environmental pressure, these variations accumulate. Traits that enhance nutrient acquisition persist, while those that do not fade from the lineage. Over many generations, the cumulative effect appears dramatic, yet each step is modest when viewed in isolation. Carnivory also demonstrates convergent solutions to shared problems. Independent lineages, separated by geography and ancestry, arrived at comparable outcomes when confronted with similar nutrient limitations. Tubular leaves evolved in distant families. Adhesive surfaces appeared in unrelated genera. Suction mechanisms emerged in aquatic habitats, distinct from terrestrial bogs. Convergence underscores that evolution is shaped by constraints as much as by chance. When the environment restricts certain resources, the range of viable responses narrows, and similar forms may arise repeatedly. At the same time, carnivorous plants remain anchored in the fundamental processes that define plant life. They continue to rely on chloroplasts, vascular tissues, and rooted growth. Their traps supplement rather than replace sunlight-driven metabolism. This continuity reminds us that adaptation does not sever organisms from their origins. It layers new functions onto established frameworks, maintaining coherence while expanding capability. Through these plants, evolution appears neither chaotic nor predetermined, but responsive. Extreme stress does not automatically produce extinction. It can foster diversification when variation exists to meet the challenge. The rise of carnivory reflects the interplay between limitation and possibility, where existing biological tools are reorganized to navigate scarcity. In wetlands and forests across the globe, these species persist as quiet exemplars of adaptive refinement. Their forms embody the principle that innovation emerges from constraint, and that life, when pressed by environmental pressure, can reshape familiar structures into unexpected yet coherent solutions. The image of a food chain often appears simple, a linear ascent from plant to herbivore to predator, each level dependent upon the one beneath it. Yet, in practice, ecological relationships resemble interwoven networks rather than straight lines. Nutrients circulate through multiple pathways, and organisms occupy roles that shift depending on context. Carnivorous plants occupy one of these intersections, complicating the boundaries once drawn between producer and predator. At their foundation, these plants remain primary producers. Chloroplasts capture photons, carbon dioxide is fixed into sugars, and chemical energy is stored within plant tissues. The trophic base of their metabolism is unchanged. Light remains the ultimate source of energy, and photosynthesis continues to fuel growth, respiration, and reproduction. Carnivory does not invert this hierarchy, it overlays an additional channel through which nutrients are acquired. What shifts is not the origin of energy, but the route by which certain elements enter plant bodies. Nitrogen and phosphorus, typically obtained from soil through root absorption, are partially supplemented by captured organisms. The plant still manufactures its own organic compounds using sunlight, yet it integrates animal-derived nutrients into its biochemical pathways. This dual strategy reveals that ecological categories are more flexible than rigid diagrams suggest. Food webs are fluid systems in which matter and energy move through diverse and sometimes unexpected connections. An insect that feeds upon nectar may later become nutrient input for the same plant species. Microbial communities within traps participate in decomposition, linking additional trophic levels into a contained environment. The resulting network blurs distinctions between producer and consumer, without dissolving the fundamental role of photosynthesis. Predation by plants does not transform them into dominant regulators of animal populations, nor does it elevate them above their foundational trophic position. Instead, it demonstrates that primary producers can adopt supplementary strategies when environmental conditions favor such adjustments. The traditional depiction of plants as entirely passive recipients of soil nutrients underestimates the range of adaptive responses available within the plant kingdom. By integrating carnivory into their life histories, these species reveal that the food web is less a ladder and more a mesh of intersecting pathways. Energy continues to flow from sunlight into living systems, yet the direction of nutrient transfer can vary locally. A leaf may function simultaneously as a photosynthetic surface and as a site of external digestion, embodying two ecological roles within one structure. In redefining the food chain, carnivorous plants do not overturn its foundations, they illustrate its complexity. Within wetlands and forests shaped by scarcity, the boundaries between categories soften and life adjusts its strategies accordingly. The result is not contradiction, but nuance, a reminder that ecological systems accommodate variation without abandoning their core principles. In a clearing where the soil remains thin and water lingers near the surface, the traps are still open. No spectacle announces their presence. They wait among moss and low grasses, integrated into landscapes that have changed shape many times since their lineage first altered course. Seasons pass over them in cycles of warmth and dormancy, yet the underlying strategy remains steady. Modern carnivorous plants continue their patient exchange with the small creatures that wander too close. During brighter months, traps form in greater number, their surfaces fresh and receptive. In colder or drier intervals, growth slows. Some species retreat into compact buds or subterranean structures, conserving energy until conditions improve. The rhythm is not frantic. It follows temperature, moisture, and light, aligning metabolic demand with environmental opportunity. Trapping itself unfolds without urgency. A droplet glistens, a chamber fills quietly with fluid, a hinged leaf rests open until contact sets internal signals in motion. After closure, digestion proceeds over days, sometimes longer, as enzymes diffuse through captured tissue and dissolved nutrients are absorbed. There is no chase, no pursuit across distance. The encounter depends on proximity and chance, refined by structural design. These species have persisted through shifting continents and fluctuating climates. Fossil traces and molecular timelines indicate that their carnivorous lineages extend back tens of millions of years, surviving warming phases and cooling intervals, expansions of wetlands, and their retreat. Their habitats may contract or fragment, yet pockets endure where nutrient limitation still favors this dual strategy of photosynthesis and predation. Even now, in scattered bogs and tropical marshes, the quiet continuation unfolds. A single plant may capture only a modest number of prey across a season. The cumulative gain is measured not in abundance but in sufficiency, enough nitrogen or phosphorus to support flowering, seed production, and another generation. Slow growth becomes sustainable growth under the constraints that shaped them. Patience defines their presence. Leaves may function for weeks before senescing, each trap a temporary organ devoted to acquisition. When efficiency declines, new traps replace old ones, and the cycle resumes. The process lacks drama, yet embodies persistence, an evolutionary solution maintained not by rapid escalation, but by steady refinement. The landscape surrounding them may appear ordinary, yet beneath that stillness lies an ongoing exchange between plant and animal. Carnivorous plants today do not signal a dramatic upheaval in ecological order. They represent continuity, an enduring adaptation carried forward through immense spans of time. In their restrained movements and measured digestion, the ancient shift in strategy remains alive, sustained by light above and scarcity below. Long before any human word existed to describe it, the shift had already occurred. No declaration marked the moment. No boundary was shattered in a single stroke. What unfolded across wetlands and ancient soils was not rebellion, but adjustment, a gradual rebalancing in response to conditions that offered little margin for excess. The era when certain plants began digesting animal tissue was not born from aggression, it emerged from limitation. In landscapes where essential nutrients were persistently scarce, survival favored those individuals capable of drawing elements from unconventional sources. Scarcity did not provoke violence, it shaped innovation. Structures originally meant for other purposes became modified, refined, and eventually specialized until leaves themselves participated in the quiet chemistry of digestion. Throughout this long transformation, the fundamental architecture of life remained intact. Sunlight continued to drive photosynthesis. Carbon moved through familiar pathways. Roots still anchored plants to their substrates. What changed was the manner in which nitrogen and phosphorus entered the system. A supplemental route formed, integrated seamlessly into existing metabolism. Evolution did not overturn prior rules, it expanded them. Nature does not violate its own principles, it adjusts within them. When environments constrain one pathway, alternative strategies may arise from the same biological materials. Enzymes already present in ancestral tissues can be duplicated and repurposed. Cellular signaling networks can be redirected toward new functions. The resulting forms appear extraordinary, yet they are assembled from ordinary components, rearranged under persistent pressure. The food web, once imagined as rigid and directional, reveals itself as more adaptable than simplified diagrams suggest. Primary producers remain the foundation, yet their roles are not confined to a single mode of nutrient acquisition. Energy still originates from light, flowing outward through ecosystems. Carnivorous plants did not ascend the trophic structure. They diversified their participation within it. The base of the system held steady even as certain branches intertwined in unexpected ways. What seems improbable at first glance often reflects a prolonged response to constraint. Given enough time and sufficient pressure, small variations accumulate. A marginal gain in nutrient uptake becomes a selective advantage. Over generations, that advantage solidifies into structure and behavior. The improbable becomes stable, not through sudden transformation, but through repeated filtering by environmental realities. Across millions of years, these lineages endured by aligning physiology with circumstance. They illustrate a broader principle threaded through Earth's history. Adaptation arises not from defiance, but from necessity. When resources narrow and competition intensifies, life explores the edges of possibility using materials already at hand. In the end, the age when plants incorporated flesh into their metabolism was neither anomaly nor upheaval. It was an expression of evolutionary inevitability, unfolding quietly within the same natural laws that shaped forests, oceans, and grasslands. Scarcity set the conditions, selection did the rest.